Welcome everybody to another Striatech interview. My name is Thomas Münch and I'm one of the founders of Striatech. Today I'm very happy to welcome Dr. Jeff Jamison. Jeff is the director for In Vivo Pharmacology at Experimentica, a preclinical contract research organization specializing in ocular research. But the main reason why Jeff joins us today is that he has developed AcuiSee. AcuiSee is an instrument to measure visual acuity of animals, and this is what we want to talk about today. Jeff, thank you for joining us.
Thank you for having me, Thomas.
Jeff, I'm very excited to announce today our collaboration. Striatech is now offering AcuiSee to customers worldwide, increasing the possibilities to measure vision in experimental animals. Jeff, maybe you can briefly explain the idea behind AcuiSee.
Sure. The AcuiSee device is a specialized operant chamber designed for vision scientists in mind. We take a lot of the requirements for a behavioral psychologist and kind of build it into the software. And we train animals to orient on a visual target, and then we can make that target more difficult to discriminate, similar to you or I reading an eye chart. We start with training the animal to discriminate a very easy target, such as the large E, and then we can, once trained, make that target more difficult and get to thresholds of vision.
Yeah, and can you explain a bit more of how that actually works, how that chamber looks like, or what's going on there?
Absolutely. So the chamber is a relatively light-tight, relatively soundproof chamber. We have a small circulating fan that allows the animal air exchange for the animal, and it also provides a little bit of white noise, and so that they're not distracted from other laboratory noises. The technician places the animal into the chamber, and then we have a touch screen orientated a distance away from the animal that will display images. We then train the animal automated through the software such that if they select a particular image, they'll get a reward. In our case, we happen to use a 10% sugar water solution. We can do other kinds of treats as well, but the idea is to get the animal to associate that image with a positive feedback. And then once the animal is trained to know that task, and they know that they'll get a treat from selecting it, we can then make that task more difficult, or make that image more difficult to see. And again, like I said, get to threshold. So we not only can test things like spatial resolution or visual acuity, like in the letters in an eye chart, but also things like contrast sensitivity or discrimination, color vision, and other aspects of moving vision as well.
Mm-hmm. Yeah, so as of course you are aware, we at Striatech have developed the Optodrum, which is also a device to measure visual abilities in animals. Correct. And the Optodrum takes advantage of the optomotor reflex, and this reflex makes the animal follow movement in its surrounding. And one can show rotating stripe patterns, and then when one makes these stripes more difficult to see, meaning one makes them finer or one reduces the contrast, then eventually the animal will not follow those stripes anymore, and this is how you find the visual threshold of the animal. And with our Optodrum, this process is fully automated and objective. And one big advantage of this approach is that there is no training required, because it is a reflex, and animals can be measured right after eye opening because there's no training required.
Absolutely.
Yeah. So how does that compare to the AcuiSee?
Well, one of the things that Optodrum really brought to the field of the optokinetic response, or this optomotor response, was the fact that you guys were able to automate this system. Mm-hmm. One of the big reasons why I started to develop AcuiSee was the the early tests for optomotor response required a technician to sit there and stare at the animal the entire time. And that was both time consuming, and there is some, intra-technician variability and some other problems with that system. So, we too started to develop our device and wanted it to be fully automated for the technicians. And so, we also wanted to make it so that you did not have to have a behavioralist or somebody with behavioralist training on staff. So the big advantage of our device is that we can get to more aspects of vision. So not just measuring the visual acuity, being able to do color discrimination, being able to do static images. Also, even if we wanted to look at movement vision of up, down versus left, right, or doing concentric rings and looking at the distribution of ganglion cell dendrites and whatnot based on the size of the ring discrimination. So there's just more flexibility or more options available in the AcuiSee device. You do have that lead time of the training, without a doubt. And since animals, especially rodents, rodents particularly are not visually oriented, it does require a little bit more time to train rodents compared to higher order species like, say, dogs or chickens, even, for that matter. But the advantage is the added features that the AcuiSee device brings.
Mm-hmm. Yeah, and maybe one of the reasons for that is that the optomotor reflex itself, it involves only subcortical areas, and one does not really measure conscious vision with our optodrome, right? So there's no decision making involved in the optomotor behavior of the animal, right? So I guess in that sense, the optodrome and AcuiSee, they very well complement each other and depending on what you're actually interested in scientifically, one or the other device might be the better choice.
Oh, absolutely. And two, the AcuiSee kind of is a very broad range vision testing device in the sense that it requires the entire visual system to be intact, from having a relatively clear cornea, through to the lens, to the vitreous, to the retina, and then all the way back to the occipital lobe. So really any alteration in vision of that, that entire visual pathway is going to be able to, will affect in essence that anything that you measure with AcuiSee. But that's very similar to what we see in patients. Usually the first line of screening in patients is the Snellian eye chart, right? Or a simple, eye exam to rule out any physical abnormalities. But once you start to see changes in the eye chart, then you can start to probe a little bit further where those problems are occurring. So again, with the AcuiSee device, it's kind of like that general test of vision and can get at a lot of different cognitive things and also retinal wiring things. But then if you start to see problems, you can use things like the optodrome or ERGs or focal ERGs or other types of things to probe a little bit more in depth into the area of interest.
Mm-hmm. Yes, absolutely. Thank you, Jeff. And this is also why I'm so happy that Striatech can now offer both the optodrome and AcuiSee to measure vision for all these different applications that are out there.
Yeah, I'm really hoping that with this partnership, as AcuiSee starts to get out there, we standardize this ability to do behavioral testing across labs. Not everybody has the ability to do this operant conditioning in a lot of visual labs. I mean, operant chambers are used throughout brain research and a lot of other areas, but I'm really hoping that by making this specialized box amenable for vision scientists and making it specifically with the vision scientist in mind, we'll be able to open up more of this cognitive and behavioral training that's been available to our colleagues in other fields to be able to start doing that kind of testing in our animals for vision.
Mm-hmm. Fantastic. Thank you so much, Jeff, for being here today. It was a pleasure talking to you.
Thank you.
And you're welcome.
It's been great talking to you as well.
More information about measuring vision, both with the optodrome and with AcuiSee, can be found on our website, stria.tech. Thank you all for watching and take care.
Thank you.